Welcome to PG Pathshala paper 11, and today we are talking about module 24, which is a continuation of module 23 about the receptor ligand interaction, and this focuses more on enzyme coupled cell surface receptor and the concept of agonist and antagonist. And so this is part 2 of the receptor ligand interaction. This content is written by Dr. S. A. Raju, who works at ICMR, National Institute of Pathology, Safdarjung Hospital, New Delhi. And I'm talking on behalf of him. I'm Dr. Rubidhar and I work as a DBT Biocare Fellow, AIMS, New Delhi. The following objectives are covered in this module: enzyme-coupled cell surface receptors, activation of receptor tyrosine kinases, activation of cytokines receptor, activation of receptor serine threonine kinases, clinical relevance of the receptor tyrosine kinases, and the concept of agonist and antagonist. So, now starting to elaborate on enzyme coupled cell surface receptors. These receptors were recognized initially through their role in responses to extracellular signal proteins that promote the growth, proliferation, differentiation, or survival of cells in animal tissues. Enzyme-linked receptors also mediate direct, rapid effects on the cytoskeleton, controlling the way a cell moves and changes its shape. Abnormalities of signaling through enzyme-linked receptors were observed in disorders of cell proliferation, differentiation, survival, migration as fundamental events, and this give rise to cancer. Now, enzyme-coupled receptors transmit signals into cell directly or indirectly utilizing protein kinases. They may be divided into groups based on the amino acids which they phosphorylate. Broadly, kinases that phosphorylate tyrosine are called the receptor tyrosine kinases, which are the cytokine receptors. Kinases that phosphorylate serine or threonine are called the receptor serine threonine kinases and cytosolic serine threonine kinases. Now, giving a more elaborate detail of receptor protein kinases, tyrosine receptor kinases are the predominant members of this class. There are about 90 protein tyrosine kinases in the human genome. They phosphorylate specific tyrosine residues on target proteins. That phosphorylated proteins can then activate one or more signaling pathways. There are two broad classes of receptors that activate tyrosine kinases. Number one, those in which intrinsic tyrosine kinase activity is absent. Cytokine receptors are one such class of receptors which are bound to the JAK kinases in the cytosol that affect the downstream signaling molecules. Number two, those with intrinsic tyrosine kinase activity, which are called the receptor tyrosine kinases or the RTKs. What are RTKs and how do they signal? Now, receptor tyrosine kinase consists of three common domains: an extracellular domain containing a ligand binding site, a transmembrane alpha helix, and a cytosolic domain with protein tyrosine kinase activity, which consists of the activation loop. Binding of ligand induces a conformation change in the N-terminal region of a receptor, causes the extracellular domain to dimerize. This causes the transmembrane domains to diffuse laterally, bringing the cytoplasmic domains into juxtaposition. The stabilization of contacts between the C-terminal cytosolic domain, causes a change in conformation that activates the kinase activity. This we can see in figure 4. Some RTK receptors such as fibroblast growth factor receptor are dimeric receptors. They are stabilized and activated upon binding of two FGF ligands simultaneously to the receptor. Other RTKs such as the insulin receptor form disulfide linked dimers without a ligand. Binding of insulin, which is a ligand, alters its conformation and receptor kinase becomes activated. The kinase domain of RTKs phosphorylate a particular tyrosine residue in the activation loop of other of the subunits in cytosolic domain, leading to a conformational change in the activation loop, which enhances the kinase activity of the cytosolic kinase domain. Activated kinase domain phosphorylates additional tyrosine residues present in the cytosolic domain of the receptor, which upon phosphorylation serves as binding sites for signal transduction proteins such as SH2, and also they can phosphorylate other target proteins, thus activating downstream targets. Now in figure 4 we can see the elaboration about how the receptor tyrosine kinase signaling works. GRB2 is one adapter protein that often facilitates RTK activation to downstream targets. GRB2 binds to phosphotyrosine residues on activated RTKs with its SH2 domain on one side and binds SOS proteins with its SH3 domain on the other side, which in turn can activate RAS GDP to RAS GTP with its guanine nucleotide exchange factor activity, as shown in figure 5. Activation of RAS leads to activate cascade of a protein kinase RAS, RAF, MEK, MAP, ERK, MAP kinases. ERK phosphorylates many transcription factors that mediate cellular response. STAT family transcription factors, another set of SH2 domain containing proteins which act downstream to cytokine receptors and some other RTKs. Now EGF signaling. RTKs can function as homo and heterodimeric oligomers as well. Epidermal growth factor receptors are one such class of RTKs. There are 4 classes of EGF receptors in humans. Her human EGFR as 1, 2, 3 and 4 respectively. In HER1 and HER4 receptor binding of ligand such as EGF, EREG, HBEGF, TGF alpha to the extracellular domain leads to homodimerization. HER2 does not bind to any ligand. It can signal only by forming hetero complexes with ligand bound HER1, HER3 or HER4. Thus, HER2 facilitates signaling by all EGF family membranes. Regulation of the phosphoinositide signaling by RTKs. Now, many RTKs and cytokine receptors can initiate the inositol triphosphate or the diacylglycerol DAG signaling pathway by activating phospholipase C or the PLC, a different PLC isoform than the one activated by the G protein coupled receptors. Activated RTKs and cytokine receptors also can initiate another phosphoinositide pathway by binding to PI3 kinase, thereby allowing the enzyme access to its membrane bound phosphoinositide substrates which then become phosphorylated at the 3 position. Signaling via the PI3 kinase pathway is terminated by the P10 phosphatase, which hydrolyzes the 3-phosphate in PI3 phosphatates. Loss of P10, a tumor suppressor gene, is frequently seen in several human tumors, which promotes cell survival and proliferation. Now, what is the clinical relevance of RTKs? Mutations that affect RTK signaling often lead to cell formation transformation, which is seen in several types of malignancies. These mutations affect RTKs or components of downstream pathways such as RAS, BRAF, MAP kinases, PI3 and AKT. This results in increased cell proliferation, survival, invasion and metastasis. For instance, mutations affecting RTKs like RAS, BRAF, PI3 and AKT are common in perpetuating the malignancy of several types of cancers and from different tissue origin. Mutations in the HER1 EGF receptor that prevents it from being incorporated into coated pits makes it resistant to ligand-induced endocytosis. Similarly, point mutations in FGFR1, 2 and 3 lead to the increase of receptors or constitutive activators in cancers such as prostate, bladder, breast, brain and lung. Several RTK antagonists are used for therapy of cancers and some are in clinical trials, which are elaborated in Table 1. So, Table 1 depicts examples of RTK targeted molecular cancer therapies being used clinically. And so, this will give us an idea about the common drugs which are being used and for against which receptor. So, as for example, trastuzumab, which is used against HER2 receptors in breast and gastric cancer. Cetuximab, which is against EGFR and is used in colorectal cancer. Panitumumab is also against EGFR and is used in colorectal cancer with wild type KRAS. Bevacizumab, which is against the EGF receptor and is used in colorectal carcinoma. Rituximab, which is used for CD20 and is used for B cell lymphomas. Pertuzumab is against again HER2 for breast cancer. Now, tyrosine and which these are all the inhibitors. So, now some reverse tyrosine kinase, we will talk about the inhibitors. Now, the imatinib, which is inhibits the c-KIT, PDFGR, EGFR, the gefitinib, which inhibits EGFR in non-cell small lung carcinoma. Also, ertolumib, which also works against EGFR, lapatinib, which against EGFR HER2 in breast cancer. Now, switching gears from RTKs, we will talk about the cytokine receptors. Cytokine receptors do not possess intrinsic enzymes activity, rather they utilize the JAK kinase, which is the Janus kinase, that are tightly bound to the cytosolic to mediate the response with their kinase activity. Some of the example of this family of receptor include the interleukin receptors, erythropoietin receptor, DMCSF receptor, CD30, CD40 and of all this category of the receptor. Cytokine receptors such as the EPO receptor are homodimers in the absence of ligand. Some are monomers which dimerize subsequent to ligand binding similar to RTKs. Ligand binding to the extracellular domain triggers a conformational change in the JAKs such that they can phosphorylate each other on a critical tyrosine in a region called the activation loop of the other monomer similar to the RTCase. Now, as shown in figure 6, phosphorylation of the activation loop leads to a conformational change in the kinase that enhances kinase activity of the JAK kinase. Activated JAK kinases phosphorylate several tyrosine residues on the cytosolic domain of the cytokine receptors. The phosphorylated sites on the cytokine receptors serve as binding sites for STAT transcription factor having an SH2 domain. Phosphorylated STATs dissociate spontaneously from the receptor and two phosphorylated STAT proteins form a homodimer. This induces a conformational change in the STATs that expose their nuclear localization signal, which is called the NLS, and hence the STAT dimers move into the nucleus where they bind to specific enhancers or promoters controlling target genes and thus alter their gene expression. STAT transcription factors are often used by several of the cytokine receptors to activate the downstream signaling pathways. However, some of them act through other signaling transducers such used by RTCase, where they transmit signal by activating RAS, phospholipase 3, PI3 kinase as described in the as we have already discussed. And figure as you can see in the figure showing the cytokine receptor activation and how the signaling take place. Switching gears to the receptor serine threonine kinases or called the RSKs. Receptor serine threonine kinases are transmembrane proteins and are characterized by extracellular ligand binding domain and cytoplasmic serine threonine kinase domains. All RSKs of the human genome act as signaling receptors for membranes of the transforming growth factor TGF-beta superfamily of secreted polypeptides. Ligands for these receptors include the TGF-beta superfamily, which comprises the TGF-1 to 3 isoforms, activins, inhibins, nodal and lefty, BMP, which is bone morphogen protein family. RSKs are known to play major role in multiple physiological and pathological processes of metazoa of life, including early embryogenesis, adult tissue homeostasis, atherosclerosis, tissue fibrosis, and cancer. There are two classes of TGF-beta receptors which are involved in signal transduction through membrane, type 1 and type 2, which are structurally similar and they form homodimers. Different TGF-beta ligands bind to characteristic combinations of type 1 and type 2 receptors. Cell surface proteoglycan, which binds and concentrates TGF-beta ligand molecules near the cell surface, facilitating their binding to type 2 receptors. Type 2 receptors exhibit constitutive kinase activity, hence it is active even in the absence of a bound ligand. The binding of a ligand to the type 2 receptor creates a receptor ligand combination that has high affinity for the type 1 receptor, thus inducing the formation of a tetrameric complex containing two copies each of R1 and R2, in which the type 2 receptor phosphorylates the type 1 receptor. The type 2 receptor phosphorylates serine and threonine residues in a highly conserved sequence of the type 1 receptor subunit adjacent to the cytosolic face of the plasma membrane, thereby activating the kinase activity of type 1 subunit as shown in figure 7. The activated type 1 receptor phosphorylates a latent transcription regulator member of the cytosolic SMADs family, TGF-beta activin receptors, which are the type 1 receptor, phosphorylate SMAD2 or SMAD3, whereas activated BMP receptor phosphorylate SMAD1, SMAD5 or SMAD8. Once one of these receptor activated SMADs are phosphorylated, they dissociate from the receptor and bind to SMAD4, also called a co-SMAD, which can form a complex with any of the 5 R SMADs. The SMAD complex then translocates into the nucleus, where it associates with other transcription regulators and controls the transcription of specific target genes. Now with this we end on the receptor function and how all the tyrosine and serine kinase receptors function. We're talking about the concept of agonist and antagonist. Receptors mediate several vital biological processes in cells, alteration in receptor expression or protein structure that constitutively activate them lead to various pathological manifestations. Hence, ability to inhibit or activate a receptor serves as tools to treat various diseases, as we already discussed in table 1. So, what are agonists? An agonist is a chemical that binds to a receptor and activates the receptor to produce a biological response. For example, isoproterenol, which mimics the action of adrenaline at adrenoreceptors used for the treatment of bradycardia, which is the slow heart rate or the heart block. Agents like buprenorphine are used to treat opiate dependence. They produce milder effects on the opioid receptor with lower dependence. Now switching gears to antagonists. So what are antagonists? An antagonist is a molecule that blocks a receptor and dampens a biological response. An antagonist blocks the action of the agonist. So it's acting like as a inhibitor. Examples: memantine, used in the treatment of Alzheimer's disease, is an uncompetitive antagonist of the NMDA receptor. Buprenorphine, a partial agonist of the opioid receptor, binds with weak morphine-like activity and is used clinically as an analgesic in pain management. Now, just to summarize this module 24, we discussed about enzyme-coupled receptors transmit signals into cell directly or indirectly utilizing protein kinases. They may be divided into groups based on the amino acid which has been phosphorylated. Kinases that phosphorylate tyrosine are called the RTKs, receptor tyrosine kinases, and the cytokine receptor. Kinases that phosphorylate serine threonine are called the receptor serine threonine kinases or the RSKs. RTKs consist of 3 common domains: an extracellular domain containing a ligand binding site, a transmembrane alpha helix, and a cytosolic domain with protein tyrosine kinase activity, which consists of the activation loop. Many RTKs and cytokine receptor can initiate the IP3 and the DAG signaling, as we call inositide triphosphate or the diacylglycerol signaling, by activating phospholipase C, which is also a different type of phospholipase C, which is activated by the G protein coupled receptor. Mutations that affect RTK signaling often lead to cell transformation, which is seen in several types of malignancies. For example, mutations in RAS, BRAF, MAP kinases, AKT and PI3 kinases. All RSKs of the human genome act as signaling receptors for members of the transforming growth factor TGF-beta superfamily. The activated type 1 receptor phosphorylates a latent transcription regulator member of the cytoplasmic SMAD family, and which then translocates the signal to the nucleus and activates the downstream transcription. Now we also discussed about agonists and antagonists. So agonists are chemicals that bind to receptor and activates the receptor to produce a biological response. On the other hand, antagonist is a molecule that blocks the receptor and dampens a biological response, and usually it is acting against the agonist. So with this we come to the end of the module 24, and thank you so much for your attention.